Hello, welcome to today's lecture. We'll be doing neurotransmission to muscles. So let's get started. So the corticospinal tract, which is from the brain, it synapses with the alpha motor neuron, which synapses with the skeletal muscle, where it stimulates that skeletal muscle. So it releases neurotransmitters such as acetylcholine into the synaptic cleft, which is right here, right? And these acetylcholine neurotransmitters from the corticospinal tract binds to the acetylcholine receptors of the alpha motor neuron. So this is the acetylcholine receptor, which results in influx of sodium ions and efflux of potassium ions. So in my previous video of action potential, we have learned that influx means movement of positive ions into a cell, and efflux is movement of positive ions outside of the cell. Okay, so there's an influx of sodium ions and efflux of potassium ions in the postsynaptic cleft. You see, this is the presynaptic cleft because neurotransmitters are going out, and this is the postsynaptic cleft where neurotransmitters are being binded to the receptors, causing the neuron or the axon hillock to depolarize. Okay, so the neurotransmitter acetylcholine binds to a neurotransmitter receptor, and this causes influx of sodium ions and an efflux of potassium ions. Now, this, of course, depolarizes the cell, right? Or the axon hillock. Now, these sodium ions move along the axon hillock, and as you learned in action potential, it opens up voltage-gated sodium ion channels, right? Because they are voltage sensitive. So it opens the voltage-gated sodium ion channels, further depolarizing the axon hillock. So we can say that these are the voltage-gated sodium ion channels, and they further depolarize the axon hillock. And this causes voltage-gated calcium ion channels to open, causing an influx of Ca2 plus ions. So this is that voltage-gated calcium ion channels. So we learned that because of that further depolarization, this affects that voltage-gated calcium ion channels so that it can influx into the cell. Influx of means is a movement of positive ions into the cell. Okay, so let's just draw that there's an influx of calcium ions because, as I said, because of these sodium ions. Now, this causes neurotransmitters, vesicles within the alpha motor neuron, right here, to fuse to the cell membrane and enter the cell membrane. Okay, so the influx of calcium ions causes neurotransmitter vesicles. I'll just draw the vesicles right here. The influx of calcium ions causes neurotransmitters vesicles to bind to the cell membrane, releasing that neurotransmitter into the synaptic cleft. Okay, so let's review this process. Corticospinal tract, which is from the brain, it releases acetylcholine into the synaptic cleft, into the postsynaptic cleft of the alpha motor neuron, and it binds into the receptors of acetylcholine, which causes an influx of sodium ions and efflux of potassium ions. This causes depolarization, right? Depolarization. And we learned an action potential. Because of this small depolarization of sodium ions, it triggers a voltage-gated sodium ion channels. And this causes further depolarization of, it becomes more positive, right? Because there's influx of sodium ions, it becomes more positive, meaning further depolarization. And this triggers the voltage-gated calcium ion channels. And since it's open, calcium ions can enter the axon hillock, and this causes neurotransmitters vesicles to bind to the cell membrane, releasing its neurotransmitters, which in this case is acetylcholine. So, neurotransmitters vesicles containing acetylcholine, right? These releases ACH into the synaptic cleft. ACH is acetylcholine, okay? That's just a short form. So, and these acetylcholine or ACH bind to the nicotine ACH receptor. That is the NACHR, okay? So let's just draw that. These acetylcholine binds to the nicotinic ACH receptors. And this is almost the same as the ones that are here. It causes an influx of sodium ions and efflux of potassium ions. Now, since there's an influx of sodium ions, it causes depolarization, right? And again, in our action potential lecture, this depolarization, this small depolarization causes a small change in the voltage or membrane potential, right? And that triggers the voltage gated sodium ion channels. So everywhere is the same concept. It's just a ligand gated channel. There's a ligand gated channel since it's detecting the neurotransmitters, right? Remember in my action potential lecture, we said that ligand gated channels, they detect second messengers, hormones, and neurotransmitters, right? And this is the neurotransmitter. It detects the neurotransmitter, causes a small depolarization, which is the influx of sodium ions, triggers the voltage gated sodium ion channels, causing further depolarization. Okay, I hope you understood. And this causes further depolarization. So let's just write that. This is that further depolarization. Now, there's something called as T-tubules. What are T-tubules? These are deep invaginations of the sarcolemma. So what is a sarcolemma? The sarcolemma is the muscle's cell membrane. Okay, you could see it highlighted in green, right? This is the muscle cell membrane, and this is the deep invagination. Deep invagination, and this region here is called a T-tubule. Okay, and in this T-tubule, there's something called an L-type Ca2 plus channel, and this allows Ca2 plus to enter the cell. Now, I said that because of that small depolarization, it triggers the voltage gated sodium ion channels, which causes further depolarization of the cell, right? Because of this depolarization, further depolarization, it triggers the L-type calcium ion channels to open, okay? So these L-type calcium ion channels open due to the depolarization, causing an influx, so let's draw it with the green again, causing an influx of calcium ions into the cell. Okay? And since there is an influx of this calcium ions, because of this L-type calcium ion channels, it mechanically interacts with a receptor called the ryanodine receptor. Okay? So let's just say that there's an influx of sodium ions because of the acetylcholine. This triggers the voltage-gated sodium ion channels to open and causes further depolarization, further influx of sodium ions, right? These sodium ions trigger the L-type calcium ion channels, and the L-type calcium ion channels open, causing the calcium ions to enter, which allows mechanical interaction. Can you see that? I drew this. There's a mechanical interaction with a receptor called the ryanodine receptor. So what is this ryanodine receptor? This receptor is attached to the sarcoplasmic reticulum of the skeletal muscle, okay? And because of this mechanical interaction, this allows calcium ions inside the sarcoplasmic reticulum to move into the cytoplasm. Okay, so this allows calcium in the sarcoplasmic reticulum to move into the cytoplasm of the skeletal muscle. So we can say the cytosolic calcium ions increase. The cytosolic calcium ions concentration increase. What does that mean? Cytosolic. Cytosolic Ca2 plus ions means concentration of calcium ions in the cytoplasm. It increases. Okay, so since there is an influx of calcium ions through this L-type calcium ion receptor, because of this, there's an interaction, mechanical interaction with a receptor called the ryanodine receptor, which allows calcium ions from the sarcoplasmic reticulum to move out into the cytoplasm. And this calcium ions bind to something called a troponin C. So this is our actins, right? This is our actin, and this is our myosin. I forgot to label it, so let's label it. And this right here is our tropomyosin. Now, the tropomyosin acts like a gate for the actin, right? It acts like a gate for the actin to prevent myosin to bind to the actin to cause muscle contraction. So, as I said, there's an increase in cytosolic Ca2 plus ions, right? This binds, because of this increase, this binds to something called a troponin C. Troponin C, as you can see, is binded to the tropomyosin. And once the calcium ions bind to the troponin C, it moves the tropomyosin away from the actin, allowing the myosin to bind to the actin, causing muscle contraction. So the cytosolic calcium ions binds to troponin C, and the troponin C moves with that calcium ion that is binded to it, moves the tropomyosin from the actin, allowing myosin to bind to the actin, and cause muscle contraction. Okay, so it's very simple, very simple. This concept right here is basically action potential, right? And this is what is new in muscle contraction. So a ligand-gated channel, which is the nicotinic acetylcholine receptor, it binds with acetylcholine, incoming acetylcholine. It allows influx of sodium ions, which causes that small depolarization, right? And we know, since this is depolarization, it requires enough nicotinic acetylcholine receptors to open to reach the threshold, right? Remember our action potential lecture? It requires a minimum. This is not the diagram for skeletal muscle, but this we're just using an example. It requires enough ligand-gated channels to open to reach the threshold, right? After it reaches the threshold, the voltage-gated sodium ion channels open to cause further depolarization, right? So once there's an enough influx of sodium ions, it causes the voltage-gated sodium ion channels to open, causing further depolarization, further influx of sodium ions. This triggers the L-type Ca2 plus channels. Okay, this triggers the L-type Ca2 plus channels, allowing Ca2 plus ions to enter the skeletal muscle cell. Okay, because of this influx of calcium ions, there is a mechanical interaction with this L-type Ca2 plus channel. There's a mechanical interaction with the receptor called the ryanodin receptor, which is in the membrane of the sarcoplasmic reticulum. And because of this mechanical interaction, this allows the calcium ions, which is stored in the sarcoplasmic reticulum, to move out of the sarcoplasmic reticulum, or the SR, into the cytoplasm. And this calcium ions, which are released, it binds to the troponin C, moving away the tropomyosin from the actin, allowing myosin to bind to the actin to cause muscle contraction. So that's why all of this is needed for muscle contraction. So the signal from the brain, which is the corticospinal tract, it synapses with an alpha motor neuron. That signal is transmitted by acetylcholine from neuron to neuron, and that causes depolarization of the neuron. And because of that, there's an influx of calcium ions, right? Because of the calcium ions, remember that it causes neurotransmitter vesicles to bind to the presynaptic cell membrane, right? To the presynaptic cell membrane. And this causes a release of acetylcholine. Again, this acetylcholine binds, which is the same here. Acetylcholine binds to a ligand gated channel, causing an influx of sodium ions, and this triggers the voltage-gated sodium ion channels because it reaches a threshold, causing further depolarization, and this triggers the L-type calcium ion channels, causing an influx of calcium ions. And because of this, there's a mechanical interaction with a receptor called the ryanodin receptor, which is in the membrane of the sarcoplasmic reticulum, right? This causes the calcium ions in the sarcoplasmic reticulum to move out of the SR into the cytoplasm, right? And this calcium ions can bind to the troponin C, which moves the tropomyosin. The tropomyosin, I spelled tropomyosin wrong here. Tropomyosin away from the actin, allowing myosin to bind to the actin, causing muscle contraction. So yeah, I wrote the points in point form from 1 to 4, and here's 1 to 8, 1 to 9 actually, from the corticospinal tract to the alpha motor neuron, which is here. I explained it in point form, and the alpha motor neuron, how it stimulates the muscle. It's like a story, so you can just read through it. I'm not going to explain it again, point by point, because it would be my third time explaining it. So yeah, I posted these notes in the Google Drive folder, which will be in the description and the comment section. And if you have any questions, you can just ask them in this YouTube video in the comment section, or you can just DM me to my Instagram page if you have any personal questions regarding the lecture, a deep question where it requires a discussion. You can just DM me at dsr.manav, dsr.manav, and I'll respond. For sure, I'll respond. So yeah, I hope this lecture was helpful. Again, these notes are in the Google Drive folder, which will be in the description and the comment section. I hope this lecture helped you, and I hope you have a great day. Bye-bye.